Here we go again. Biology, Cape Unit 1, July 2021. There we go. There's a COVID here. So remember the format of this changed. There's 3 questions now instead of 6, with each module being represented by each question. So question 1. In the space below, construct an annotated diagram of the E. coli bacterium from the electron micrograph provided in figure 1. Show the magnification of your diagram. Well, you know, CXC is sometimes you could barely see the thing. The picture quality is not the best, but this was whopping 8 marks. So assuming that we wanna label at least 5 things. So you're gonna see 5 things labeled here: the cytoplasm, the cell wall, the plasma membrane, the nucleoid region, because remember, bacteria prokaryotic, which means that they don't have a true nucleus, just a region with genetic material and circular DNA and things like that. And ribosomes might be represented by some of these little dots that you're seeing here. Otherwise, I can't really see much else. Usually some of these bacteria would have a flagellum, but you can't see that, so you don't label where you can't see, right? Of course, this would have to be a drawing. You just draw you just draw something very neat, representative of what you're seeing on the diagram, and label accordingly. The cell wall, I should note here, is not the same as a plant cell wall. The cell wall here is made out of not cellulose, but something called mucin. This is the breakdown of marks that you're seeing here. So any 4 annotations above, you could also annotate mesosome, cyme capsule, plasmid, and cilia. The drawing must occupy at least half of the box shown. Clean continuous lines, no shading. You include a title, drawing of E. coli bacteria, and a magnification accurately calculated. But since you're watching this on the computer, you wouldn't really be able to do it. You wouldn't be able to do it accurately here. So if you get a physical copy of the paper, you could do that. Alright, let me look at B here. Plant and animal cells are the most common types of eukaryotic cells. Remember, eukaryotic means that they have a true nucleus. List 3 similarities between a typical animal and a typical plant cell. When they say typical, they mean undifferentiated. So they're talking about like red blood cells and phloem tube cells and things like that, right? Just your normal cells. So both cells would have mitochondria, both of them would have ribosomes, both of them would have a smooth and a rough endoplasmic reticulum, both of them would have nuclei with the nuclear envelope and the nucleolus, all that containing DNA and RNA. Both of them would have plasma membranes or cell surface membranes. Both of them would have cytoplasm. Any 3 of these really would work. Is 3 similarities, so you're good to go. The next part is outline the function of any 2 structures found only in plant cells. Well, here we go. Well, chloroplasts are the site of photosynthesis. They allow the conversion of raw materials such as water and carbon dioxide into glucose using sunlight. The cellulose cell wall prevents plant cells from bursting due to excessive turgor pressure. It has plasmodesmata, that has little gaps to allow water flow from one cell to another. You'll learn a little more about that in Unit 2 through what they call the symplast and the apoplast pathways. Lastly, starch grains are found only in plant cells, and these are energy reserves, but animals don't contain starch. They have glycogen granules. Any 2 of these with a proper description or description of the function would net your 4 marks. This question is a little confusing to me because I have no idea how this is supposed to make up. This is supposed to be 15 marks, right? And I mean, I write as much here as I know. So let me read here. Starch comprises of alpha glucose monomers linked by 1,4-alpha glycosidic bonds in amylose and periodic 1,6-alpha linkages in amylopectin. These units form a repeated linear chain in amylose and branches in amylopectin. We look and see here, well, what is what? Here we could see this one here. This is amylose. Right, so this structure here is amylose, and basically what this is made up of is a bunch of alpha glucose molecules joined together here. Now you'd ought to draw. I assume here that you could possibly draw it in this question here, and it's easy to draw. So we could draw the molecule. Here we have oxygen. Here we have a carbon. Here we have a carbon. Carbon to remember 6 carbons in glucose, and this would be an alpha glucose molecule. So far we have 1, 2, 3, 4, 5. The 6th one is going to be outside here, and this is going to be CH2OH. You'll have to know how to draw this anyway for your unit 1 syllabus. And remember, carbon must have four bonds to it, right? So this is how we remember this. So this is going to be ho. You might be like, well, why are you talking about? Well, if you look at this, this is ho. Right, so remember ho. The hydrogen and the hydroxyl, ho. Now there's an alpha glucose, right? If it was a beta, these two H and OH here would be inverted. Alright, so you see what I'm talking about? Ho. Now I make it label each of these carbons. This is carbon 1, this is carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Kind of special, he just on the outside there. So this is one alpha glucose molecule, and amylose, they're gonna get a string of these connected. So how do they connect? So if I put 2 of them side by side like this, you'll notice here, if I was to connect this hydroxyl with this hydrogen, but if you notice here, this is H2O, right? And this is gonna leave as water. And what we're going to be left with is this oxygen, this bonding to this here. So you see that this is bonding this carbon 1 to this carbon 4 of the adjacent molecule. So this is why we call this a 1,1,4 glycosidic bond, or glycosidic linkage. Glycosidic just comes from, like, glucose, sugar, and since it's an alpha, you can say 1,4 alpha, and you just draw like 2 of these and indicate that that it'll be going like that down the road, and that there's amylose. Now amylopectin, so what's different here is that it has the same linkages here, but amylopectin would also join the hydroxyl group from the CH2OH, where you're seeing here, to the OH of a branch here. I wouldn't think you have to draw that there, but you should link that there are 2 different linkages in amylopectin. There's a 1,4 and a 1,6. So back to the question here now. So that is starch, and starch consists of those 2 types of sugars, amylose and amylopectin, and the types of linkages that I see here. Cellulose, on the other hand, comprises of beta glucose monomers linked by 1,4 beta glycosidic bonds. So even though they form linear chains, just like amylose, which was over and over again, each successive monomer, a monomer is just a unit of sugar or a unit of some molecule, experiences a 180-degree inversion. Cellulose chains are also much longer and contain much more hydrogen bonds. That's why they're so strong. Even though hydrogen bonds are weak, there's a whole lot of them that you have to break in order to you could think, unravel cellulose. So it has much more structural integrity than starch. And these linear chains even form hydrogen bonds with each other. So let me show you here. So here we could see the difference between starch and cellulose. Well, this is amylose and cellulose, right? Again, you'll see that each monomer here is the same, and these are alpha glucose molecules. These here, however, are beta glucose molecules. And what is happening here is that you'll notice that each successive unit, look at where this group is, CH2OH, and look at where this is here, is inverted 180 degrees. Look at where this OH here and look at where this OH here. So it's basically flipping upside down each time. So the next time that the next one that's gonna be connected here will be this one here. This is the one that's gonna link over there. That's the one that's gonna connect there. And then this will continue going right side up, upside down, right side up, upside down, right side up. And that gives it a lot of structural integrity. Now it's not just that, these will actually be stacked one on top the other. So if you used to imagine how cellulose would be, right, you would have a bunch of them linking up one on top the other like this, going like stacked, like layers, and that makes it a lot stronger. Starch molecules are more soluble than cellulose, as starch molecules can be dissolved in warm water. They are thus more easily digested, and they can have faster interactions with enzymes, so that they could separate the polymer into the glucose monomers. Question 2. DNA and RNA are made up of polynucleotide strands. Figure 2 represents part of a DNA molecule. Here you can see part of it, and they said on the label diagram, provide labels to identify the parts of the DNA molecule labeled S, T, U, and V. Okay, so what you're seeing here is part of a DNA molecule, and you have one side going from 5' to 3', and the next side basically going the opposite, 3', 5', right? Basically inverted. So all the way to do here is label. So S now, this will be the phosphate group that's attached to it. T is going to be the sugar or deoxyribose sugar, and for RNA this will just be a ribose sugar. U is going to be a base, but in this case here they probably wanted to name the base, and that will be where guanine connects to cytosine. Think of it like the car in the garage, C and G, car in the garage. And V is thymine, because that is connected to adenine. And think of that as the apple in the tree. The apple in the tree, the car in the garage. On the diagram in figure 2, draw a circle around a section of the diagram to identify a nucleotide. So a nucleotide consists of 3 things, which would be a phosphate group, a sugar, and a nitrogenous base, which in DNA will be either adenine, cytosine, guanine, and thymine. And in RNA, you won't have thymine, you will have uracil. State how 2 nucleotides in a DNA molecule are held together. Well, this is called a phosphodiester bond, and it's by a condensation reaction. When you want mark, you just have to name it. Part 4. DNA codes for polypeptides in cells. Different types of RNA are involved in this process. Outline the roles of messenger RNA or mRNA and transfer RNA tRNA in the production of polypeptides in eukaryotic cells. To break this down here, mRNA facilitates something called transcription, tRNA facilitates something called translation. The DNA strand is going to be used as a template to build an mRNA strand, and that's why it's called transcription. This messenger RNA now is like a blueprint that will be used to build a protein. This is like the instructions to build a protein or a sequence of amino acids or a polypeptide. And in this case here, you'll see how the bases connect. A connects to U, car in the garage, C and G, C and G, A and U, and so on, right? These letters here now, well, I mean, they don't spell anything really, because they're just the bases. If you take them 3 at a time though, like UGG for example, or UUU, this here is what you call a codon or triplet codon. So if they are taken 3 at a time, this here is the instruction for a certain amino acid. So right now, this here is just an instruction, but this gets translated to build into amino acid. And tRNA helps with this translation here. So this codon, UGG, this will help to assemble an amino acid called TRP, tryptophan. If we look at this one here, UCA, this would translate to assemble an amino acid called SER, serine. So each codon there, think of it as each one is the instruction for the placement of a certain amino acid. The transfer RNA reads this blueprint and it puts that specific amino acid there. And that is how a protein is built, because all these amino acids gonna make up a polypeptide. Those polypeptides are gonna get linked together in a kind of three-dimensional way, and that's gonna build a protein. So mRNA facilitates transcription of nucleus DNA. It is mobile, which means it's actually able to come out of the nucleus and go into the cytoplasm towards the ribosomes. So it carries the RNA genetic code out of the nucleus into the ribosomes in the cytoplasm. Transfer RNA allows the binding of an anti-codon to mRNA codon. So I'll put up a little diagram just to show what that means. Carries the individual amino acids to the ribosome. So like how each code that I saw there was for a specific one, like how you had the tryptophan and the serine, that's what happened there. Eventually this is going to link with peptide bonds because it's a polypeptide chain to form polypeptides, and eventually they will fold into proteins. These proteins could be anything. It could be hormones, it could be collagen, it could be hemoglobin, it could be melanin. Sickle cell anemia is a disease caused by a change in a gene that encodes hemoglobin. Name the change in a gene which results in the production of abnormal hemoglobin in sickle cell sufferers. Well, this is called a substitution mutation. So let me go through it here. So this is your a normal DNA sequence, GAG. And this is gonna pair, G is gonna pair with C, A is gonna pair with T, G is gonna pair with C. And this here, the mRNA that's gonna be built from this, C is gonna pair with G, T is gonna pair with A, C is gonna pair with G, right? That shouldn't be a problem. And that codes for a protein here, GLU. And that codes for a protein here, GLU, glutamic acid. After a point mutation though, so a point mutation or a substitution mutation, what that does is that it changes a single letter in what is supposed to be the normal sequence. So the normal is GAG, but that changes the A to a T. So from the ground of the instructions have changed because GTG, right, the complementary DNA is now going to be CAC, and that template is going to be built, the mRNA. C is going to pair with G, A is going to pair with uracil, and C is going to pair with G. So now we end up with a totally different set of instructions that the mRNA is carrying to be translated. And that's going to make a totally different amino acid named valine. And this placement, this rogue amino acid this is actually going to change the entire shape, the entire form of the protein that is supposed to be hemoglobin. And as a result, what's going to happen is that the shape of the red blood cell is going to change, and thus the function of it will also be affected. So part 2 is saying the hemoglobin that is produced in sickle cell anemia leads to sickle-shaped red blood cells under low oxygen saturation. Outline how low oxygen saturation results in a change of the shape of red blood cells. So hemoglobin is a quaternary protein, which means it's made up of multiple tertiary structures. Right, so it's comprised of 4 tertiary proteins. I'll put up a diagram so you can see what I'm talking about. So a substitution mutation changes a single nucleotide. This means that one letter has changed, right? One base pair has changed, eventually resulting in the translation of an entirely different polypeptide and protein, because instead of GLU, that amino acid, you end up with VAL. This changes the phenotypic expression of the red blood cells, affecting the curvature and ability to bind oxygen molecules. Now, typically, what the ability that hemoglobin has is that when an oxygen molecule binds to one of the 4 sites, it actually makes it a little easier for the other 3 to be able to bind oxygen. So you could say what we call the affinity for oxygen increases. So if you have 4 available spaces and one binds, it makes it easier for the other 3 to bind, and so on. So this reduces the red blood cell's ability to exhibit conformational changes. This means that the red blood cell actually folds a little bit to make it easier to bind more oxygen, and thus production of oxyhemoglobin. Alright, so let's look at C. In 2017, French scientists announced that gene therapy had been used to successfully treat a patient with sickle cell anemia. Figure 3 shows the results, which detail the patient's response to low oxygen levels after 6 and 12 months of treatment using gene therapy. They also had 5 control subjects to low oxygen levels. So what is this graph showing us here? So this bar graph here is showing us 5 different controls, and then we have the patient after 6 months of therapy and 12 months of therapy. Now the first control is the patient's mother. So the first one we can see is the patient's mother, and assuming we're reading from up here, and she has about, you could say, 25% sickle red blood cells. The control 2, right, is somebody who has no symptoms, and in low oxygen conditions they have about 15% or so. I just eyeballing this thing, right? You all could look at it properly. Controls 3, 4, and 5 are people with sickle cell anemia, and they have relatively high, almost 50% sickle red blood cells. The patient has about 50% sickle red blood cells in the beginning, but this is after 6 months of therapy, which is after 6 months of therapy, not much change happened, because you could see that the patient almost has the same percentage as the people who have sickle cell anemia. That's 3, 4, and 5. But after 12 months of therapy, basically a year, you can see that the number has significantly dropped, the percentage of sickle red blood cells. So that means that the therapy has some degree of success. So the question here is, using the data given in figure 3 on page 12, analyze the success of gene therapy treatment for sickle cell anemia. You're basically just commenting on the graph, everything that I said. So patients who receive gene therapy, there seemed to be little change that occurred after 6 months. Approximately 45 to 48% sickle cells were still there. However, after 12 months, that number reduced to approximately 25 to 28%. This was close to control 1, the parent, the patient's mother. An assumption can be made that if the therapy continues past 12 months, the percentage can be further reduced. Alright, so this was for a good 15 marks here, either 12 or 15 marks. Discuss 3 challenges which may limit the success of gene therapy as an effective treatment for diseases in modern medicine. Typically, you would use something called a restriction enzyme that are usually produced by bacteria like E. coli, for example. A restriction enzyme acts as DNA scissors to cut out a little piece of DNA that you could then use an enzyme called DNA ligase. DNA ligase is a kind of glue that you could use to attach it to a plasmid. So that's basically what you're seeing here. So this protective gene, right, is being attached to a plasmid, and this plasmid here that I seen in 2, which is just like something that could be taken up into a virus. So this uses a virus as a vehicle or a vector in order to transport it into a cell. So it might be like, well, why a virus? Because then virus is bad. But viruses are very good at penetrating cell membranes. They're actually very good at getting into cells. So they use viruses that mostly the harmful parts have been deactivated. However, you have a lot of risk with this because you don't know if the virus could mutate or reactivate while inside the person's body and compromise the immunity. So the virus enters the cell, and the capsule of the virus breaks down, releases the plasmid, and then the plasmid with the new gene is taken up into the nucleus of the cell, the target cell, and then it will be integrated into the cell. And hopefully I have no viral replication and things like that that would cause any illness. Of course, the technology is still new, so they haven't smoothed it out yet, so a lot of things could still happen. Now the problem with this here is that you don't know if the virus will actually head to the target cell. It might end up depositing the DNA into some other cell that don't need it. The virus itself could reactivate or mutate. And well, eventually now this new cell that the DNA has integrated into, the host cell, this cell will eventually die. So unless this cell could reproduce fast enough and make plenty of the new DNA, the whole thing is gonna be useless in the long run. So they want to know what limits the success. So 3 challenges. I'm gonna put a little detail to this. So since gene therapy tends to introduce DNA to the patient's body by the use of viral vectors, they are always high risk of infection, especially to those who are immunocompromised. Remember, the viral vectors could also mutate, right, or reactivate and start to multiply within the patient. There's been a few deaths with cystic fibrosis gene therapy that involves something like that. The viral vectors may not target enough cells to make a substantial phenotypic change or allow the modified cells to reproduce quickly enough. So you'll have, let's say you're aiming for 1,000 target cells, well, the viruses might invade other cells instead, and it might only actually penetrate 100 out of that 1,000. That is not high enough. Those cells might die out before they could start to replicate. There's also the risk of the viral vectors not targeting the cell at all. The patient's immune system may destroy the viral vectors. So if your white blood cells, your lymphocytes and things like that might actually just aim to get rid of whatever is delivering the new DNA. That's not good. The new DNA may result in unforeseen mutations or tumors in the modified tissues. They may also produce negative metabolic side effects, or they may trigger allergies in the patient. So again, unless extensive research has been done, this new DNA, which is really just supposed to change one trait or one characteristic, may end up changing multiple characteristics, or again may trigger allergies, so that they would limit its success. Question 3. Data on the relative levels of hormones, FSH or follicle stimulating hormone, and LH, luteinizing hormone, in the blood during the menstrual cycle are provided in table 1. Here you can see the table. Here we have different days of the menstrual cycle, and it goes up from day 5 to day 27. And here we have the concentrations of the hormones FSH and LH. This is the table, and this is the graph. So you could draw the graph for yourself. You'll see that I've plotted all the points. What you want to make sure is that you have a correct scale, you have a title given. See my title? You have a key or legend given. In my case here, I could have put it in 2 different colors. But what you all could do is do the 2 different types of plots. So you could use X's for ones and dots with circles for the next one. Or you could draw one as a solid line, and you could draw one as a dotted line. The next question, part 2, outline the role of the hormones FSH and LH in the maturation and release of the secondary oocyte during the menstrual cycle. The secondary oocyte is the egg. So what LH does is that there's a surge in LH. So LH stimulates the release of the secondary oocyte by facilitating what's called a positive feedback mechanism that causes a rise in estrogen. What this means is that the more LH is produced, the more FSH is produced, right? One triggers the increase of one triggers the increase of the next one. And in the end, this allows the Graafian follicle to rupture and thus be releasing the secondary oocyte. So you could actually see the spike on the graph, which you should see on your screen here. FSH allows the growth and maturation of the Graafian follicle. So you could think of that as actually increasing the size and helping with the development of the follicle. And that follicle contains the secondary oocyte. The follicle is like a little casing fit. So as FSH is released, the follicle actually releases estrogen from the cells called the Theta cells. So that is how the two of them help with maturation and release. B. There's a 4-mark question. Following the release of the secondary oocyte, fertilization normally occurs in the oviduct and implantation of the blastocyst occurs in the uterus. Outline the process of implantation of the blastocyst in the uterus, including hormonal influences. The blastocyst is like a ball of cells. So when the sperm cell fuses with the egg cell, it forms a zygote. That zygote now is going to undergo mitosis and is going to keep dividing until it forms a hollow ball of cells called a blastocyst. The blastocyst would then develop into the embryo and then into the fetus later on. Now the blastocyst contains these specialized cells called trophoblasts. And trophoblasts think of it like they are on the border of the blastocyst, the outer border, and they secrete enzymes that allow attachment of the blastocyst to the endometrium. So those enzymes, think of it like they're gonna digest a little hole or a little pocket in the endometrium. The endometrium is the uterus lining, and they're gonna fit itself snugly inside that little pocket there. So the blastocyst could actually connect to the mother's bloodstream. So when it connects now, the trophoblasts, we think of them as little tentacles, little fingers that help dig into the endometrium. I don't like to use the word dig, but help it fit or help it grip onto the endometrium. So these then mature into chorionic villi. And these villi now, what is very important about this is that this will actually connect the maternal bloodstream to the fetal bloodstream. And this would actually keep both bloodstreams separate from each other, because the maternal bloodstream would have the blood pressure would be too high for the fetus. So this would then facilitate the transport of materials like oxygen and glucose, and the removal of materials like urea and carbon dioxide. The blastocyst secretes HCG, which stands for human chorionic gonadotropin. What HCG is the hormone that is only produced during pregnancy. So when a woman is taking a pregnancy test and she has to urinate on the little stick, and the line shows up, that is actually detecting HCG. And HCG, as I said, is only present in pregnant women. So what does that hormone do? Well, HCG stimulates the release of progesterone so that menstruation does not take place during pregnancy. Of course, menstruation cannot take place during pregnancy. Well, at least a full menstrual flow cannot occur, because the uterine lining would shed, and then the embryo or the blastocyst would shed with it, which would be a spontaneous abortion. You don't want that to happen. You want the endometrium to remain thickened, right? To remain thickened, and so that it could accommodate the implantation of the embryo, and so that it could remain attached to the uterus and thus to the mother's bloodstream. HCG prevents menstruation from occurring. Okay, let's look at C. In plants, both sexual and asexual reproduction can occur. In sexual reproduction, floral structures develop into mature fruit following fertilization. A diagram illustrating a longitudinal section through the ovary of a flower is given in figure 4. And here we could see this is a ovary, and we're gonna have two parts labeled A and B. So just to note, what is A and B, right? So A is actually the ovary, and after fertilization, this ovary here becomes the fruit, and the ovary wall becomes the flesh of the fruit, what we call the pericarp. Now this inside here, this is an ovule, and the ovule becomes a seed of that fruit. So state the change in each of the label structures A and B in figure 4 in relation to development from floral to fruit tissues. Well, A would become the fruit, or if there's pointing to the ovary wall, that would become the pericarp or the flesh of the fruit, and B would become the seed. Part 2. Discuss 3 advantages of asexual plant reproduction for agriculture. Include 1 limitation for each of the advantages discussed. I believe this was 12 marks. So you want to have a good description of each advantage, but then you want to have 1 limitation for each of those advantages. So asexual reproduction allows for rapid production of plant offspring. This is because you don't need pollination and you don't really need fertilization or anything like that. Asexual reproduction basically involves cuttings, grafting, where you just have one parent and you have no gametes involved. This could also involve tissue culture. So using asexual reproduction and producing a lot of plants, you would have high profits in agriculture because only one parent is required. You don't need any pollen. You don't have any mixing of alleles or anything like that. You basically get clones. Now the bad thing about this is if you get clones is that it could lead to a very limited gene pool and reduces vigor in the plants. This would make the plants very susceptible to pests and disease. Good example would be some banana plants, and those are grown from cuttings, not from seeds, because the seeds are not viable in bananas. So when one pathogen tends to affect one banana plant, it could affect a whole bunch of them because they are very genetically similar. The second point here is asexual reproduction allows for beneficial characteristics and alleles to be passed down to plant offspring. So remember, it's clones, so the same characteristics would be passed down. So there's no mixing of alleles from other species members. In agriculture, this could result in higher yields or plants that are favored for specific abiotic factors. However, this means that plants would only be able to thrive under specific conditions or soils. So an example would be like, what if you was to make a tissue culture for a plant that would only grow in acidic soil because there's only acidic soil in the area. Well, all of a sudden now if you was to plant it in alkaline soil or neutral soil, it wouldn't be able to thrive as well. Again, this limits its survivability, but once you have a stable ecosystem or at least a controlled ecosystem, you would be able to produce a lot of plants in a short span of time. But it would have to probably be maintained within that contained or controlled ecosystem, like for example a greenhouse. Third point, asexual reproduction such as vegetative propagation can easily be manipulated and scaled in agricultural operations. However, this whole process here of cutting and grafting and so on, this could be expensive, and it requires at least a somewhat high amount of expertise to maintain environmental sterility, especially with tissue culture. Tissue culture, you use small little plantlets, and those are very susceptible to bacteria and fungi. And of course, if it's not in a sterile environment, all of them would die. The whole thing would be for naught. So it is expensive. It requires a lot of equipment, a lot of expertise to make sure that it is done properly. Good example would be tissue culture, as I said. And upon fourth point here, asexual reproduction is much more time efficient than sexual reproduction as no pollination mechanisms are required. This would result in a high quantity of produce for sale, resulting in profits. And then you could say, well, in this case here, you have the same disadvantage as I said before, environmental sterility. You need to maintain a controlled environment that requires money and expertise and modern equipment. So you can't just, gear up and say you want to do it. You have to make sure that you have everything in place. And that day is June 2021. See you all here. Laters.